So the advent of XFELs and to a limited extent the ultra-low emittance synchrotrons already enabled serial femtosecond crystallography, time-resolved single femtosecond crystallography, and many other kinds of experiments. We're going to hear later about the new light sources that are coming online around the world that will have even more exciting characteristics. And it seems an appropriate moment to rethink the whole workflow of how we handle crystals. Ultra-rapid data collection is only one aspect of this, but there are really a lot of others. If you examine the workflow involved in crystallography, basically there are three stages. There's crystal growth and optimization, crystal mounting, handling and shipping, and diffraction data collection. And these components have very different weights in different kinds of laboratories. The firms that were associated with the IMCA beamline may have hundreds of crystals of the same protein, but with differing ligands in each one to study drug targets. And so the actual problem of crystal growth and optimization was solved a long time ago. On the other side, there are more and more difficult crystallization targets, and line one becomes very important. The drug companies are really eager to do rapid diffraction data collection, and that's important not only for throughput, but for time-resolved studies. And I'd like to talk a little bit about some vignettes of new technologies that pertain to each of these things, and you'll hear more about some of them today in much more detail. But there's a lot going on. It's actually an exciting time to just sit there and read the literature and be overwhelmed with how clever people are being. But before I actually start on that part, let me do a prequel. A lot of the technology we're talking about was developed for use at X-ray free electron lasers, and particularly the LCLS. And essentially all of the users of the LCLS have been not-for-profits. Academic proposals or not-for-profit research institution proposals are reviewed by a proposal review panel, and it's a very competitive environment. What do you do if you're a drug company who wants to do a proprietary project? Well, companies negotiate directly with LCLS Director Michael Dunn. This is his email. And can it happen? Well, almost. So this is all I know about the most advanced corporate negotiations with the LCLS. This is a nameless large company. And you can read for yourself that the process of discussion went really well. Michael was receptive to their ideas. And of course, the lawyers are going back and forth about the user agreement. But it's interesting to look at the bottom of the second paragraph. The cost is very high. The cost recovery rate for the LCLS has been calculated at something like $40,000 an hour. And this is a very expensive experiment, as it says here, even for us, whoever us may be. But there are mechanisms for companies to try out the LCLS at much lower cost. They could, for example, apply for a much shorter time to just do a trial to see if their crystals diffract. That would be less expensive. They might be able to negotiate with an academic lab to help them at the end of a beamline run to do their own experiments as well. So I can imagine an experiment maybe for a third this price or a quarter this price as a trial. But it's still a serious undertaking, and I think we all agree that companies should pay their fair share, but at some level, if it becomes impossible to use the facility for projects that are actually potentially good for everybody, maybe the model is a little imperfect. So let me return now to those workflow items and start with the idea of crystallization. And what I'd like to talk to you about is some work from the work of Guillermo Calero at the University of Pittsburgh. Guillermo is one of our bio-XFEL investigators, about the use of transmission electron microscopy and looking for nanocrystals and evaluating their quality without having to go to the LCLS. If this technology really proves to be useful, it's the kind of operation that might best be carried out in a facility rather than every lab trying to have something like this on its own. The ASU cryo-EM facility will certainly have transmission microscopes as well, and could be a natural home for this. And in addition, the Halpin Woodward High Throughput Lab would certainly consider trying to expand its services if this works out. So these subsequent few slides come from Guillermo. And what happens basically is that an electron microscope grid is dipped into a drop of crystals and then rapidly stained and looked at in the electron microscope. It's a quick process. And here are some pictures of crystals, just to show you that the lattices are quite visible. Here's another group. Some of them are better than others. And they're all small. Okay? We're looking at real nanocrystal sized objects here. I can't always read these even up close, but they're tiny distances. So one of the things that the Calero lab found when they started working on these is that nanocrystals were very common. They started with 30 targets and found nanocrystals in the crystallization trials for all of these. And this is consistent with some of the ideas that were floating around at the beginning of bio-XFEL, when there was a universe of unknown nanocrystals that we've been throwing away. But not all laboratories quite agree with this observation, just to be clear. At Halpin Woodward, the lab has done a study of how often nanocrystals occur using submitted proteins that were in large part single polypeptide chain proteins and not large complexes. And what HWI lab found, Joe Luft in particular, is that in every single tray in which there were nanocrystals, these are 1,596 well trays, there were also larger crystals in other parts of the tray. So nanocrystals didn't occur uniquely very often. But I think both of these observations are largely restricted to small soluble proteins, and the stories will certainly be very different for membrane proteins and for large complexes. In any case, you get to see whether the crystals are there, and you do get to see as well something about their quality. So these are crystals, and these are optical diffraction patterns taken from masked out areas of these images. And these patterns turn out to be very well correlated with the observed X-ray patterns from the same crystals when they were examined later. So if you look here, you can see that the diffraction from the crystal is anisotropic, and sure enough, anisotropic. Very nice, rich, even diffraction pattern, high resolution. So this is a picture, again, showing more detail in these X-ray photographs compared to the optical diffraction patterns from the crystals. So this represents a kind of nice technology for having a look at your nanocrystals and having some sense of whether their patterns are going to be really strong or not without having to go to the LCLS. And indeed, to think about using these things for electron diffraction experiments without having to go to the LCLS at all. So I'd like to move on to talk a little bit about crystal delivery. And you're going to hear a lot more about that today from people who know a lot more than I do about it. But just let me list 5 methods of delivering crystals to nanocrystals to XFEL beams or to synchrotrons, in at least some cases. And the first technology for delivering crystals to the XFEL which was what proved that it would work, is a liquid jet that carries a slurry or suspension of crystals in it, and which may or may not break up into droplets depending on how far one is from the source. And it was a big success. It's used still in time-resolved studies. It takes a lot of protein and had a poor hit rate. Perhaps the most exciting development since then has been the lipidic cubic phase jet, in which a ribbon of toothpaste, which is actually lipid, moves slowly in space carrying membrane protein nanocrystals. And the speed of the jet is such that it's interrogated very frequently by the X-ray beam and is much more efficient. An analog of that has been developed by Chelsea Conrad in Petra's lab, in which the lipidic cubic phase has been replaced by a viscous aqueous phase, and makes it more suitable for soluble proteins. And then there are two related technologies in which one puts the crystal into the beam in known places. One of these is a fixed target in which one has some kind of support on which crystals are mounted, and they can be pre-located and then placed in the beam. And the other is a special case then, which we call in situ, where crystals are photographed in the container in which they are crystallized. And there's been a lot of interesting stuff going on with that too. Let me give you a few vignettes. This is again from Guillermo's lab. So this is a picture of a plastic grid. The windows are filled with mylar, and this is a picture of the Mitogen loop just for size scale. And this grid is dipped like a little shovel into a drop like this and gets covered with crystals. It's then quickly viewed in a microscope, and the grid appears on the computer screen. Here are some photographs to show how well one can see crystals of various sizes. And then they use peak finding software to create a list of where the crystals are with respect to fiducial marks on this grid. And it's used either at the LCLS or at a synchrotron to collect photographs, and it gives really good data. This is cryo. And this is a picture just to show how the crystals are visualized in different ways. This is, I think, I forgot why I put this slide in. It's simply more diffraction, but it shows how nicely the data go out to 2.5 angstroms. The virtue of this is simply that it really reduces crystal handling a lot. You get many crystals on the thing. You don't have to hunt for them. The microscope locates them, bang, bang, bang. But let's look now at a couple of other versions of fixed target methods of crystal delivery. This is from Phil Anfenrood and Dwayne Miller, and it's been used both at the synchrotron and at I-24 at Diamond, and is suitable either for room temperature or cryo. And it starts with a, forgot the dimensions of this exactly. It's a fairly large compartment that's divided into these smaller squares, and that's a blow-up of these. And each of these is divided up into the final little holes that are going to contain the crystals. And the paper uses the term features for the little holes. These are made by lithography. And you can see here, this is the actual hole in the bottom, and this is the sloping sides of the hole. This is a cross-section through it, and crystals are mounted by loading on a droplet and sucking out water from below using filter paper. It's my sense that filter paper has had a long as lifetime as anything else in all of crystallography, okay? It's technology that just seems to find new ways of it. So this is a picture of a diffraction pattern in situ from an ordinary crystallization tray and from one feature of their new tray. And you can see the very, very much decreased background. And this is an average of background azimuthally averaged to show you that there's really a very big difference between this silicon chip, which has a hole in it, and an ordinary X-ray chip. You can see here they have a special mounting to hold this device. And it looks very promising. I have to tell you, this is a very substantial paper. It has a lot of interesting experiments in it, and I'm only giving you just a gloss about what it has in it. A related technology comes from the Means group, and it also uses silicon. This is a 4 by 2.5 silicon plate that is thinned in one region to a very thin silicon membrane, which is still single crystal, okay? And this is 130 microns thick. And this is 10 microns thick. And this region over here is, again, broken up into features that are created by lithography. And in this case, not all the features are the same size, or at least the same shape, so that you have the possibility of catching a crystal that's long and thin in a hole like this, and one goes in a different orientation like that. So it's a way of improving orientational sampling by having different shaped holes, or features, even though they're roughly the same size. Here's a picture of this thing. This is a picture of how it's used. The crystals are mounted, a drop is put on, humidity stream keeps them there. The magic ingredient sucks the liquid out from the bottom. You end up with the crystals distributed across the surface of this, and then you can freeze them if you choose to do so. This is a picture of the scattering with nothing in the beam and with their little plate in the beam. And you can see that things are extremely similar, except you're seeing these few X-ray reflections from the crystalline silicon. And you know, it's my sense that these things are no worse than the gaps between tiles in a detector, and that one can, in a software sense, easily eliminate. So it's, and this, I think that maybe I'll skip this in the interest of time. This is just a picture of how the thing is mounted and used. Seems quite efficient. And this is a picture of the light microscope and a scanning EM showing how crystals occupy the different holes, and an advertisement for how good the electron density maps are. And again, this is a very substantial paper with a lot of controls and a lot of applications in it, and only giving you a hint. Finally, let me show you one last application, which is a new version of the in situ process in which crystals are photographed in the vessel in which they are grown. So this is a device which has an upper and a lower seal that can be removed easily. The actual crystallization chamber is defined by a much thinner upper film and lower film. This would be a square or rectangular thing going to the board. And this space between the upper and lower film is divided into chambers by these little adhesive spacer units. In this picture is shown an experiment which is basically vapor diffusion. This is a drop, this is a crystallization drop. But if you adjust the spacing here, you can also do hanging drops and things like that. And again, the virtue of this is that it has very low background. This is a picture of a region of one of those wells. The grid is the result of a scanning search for diffraction spots on I-24. And you can see every place that there is a bright thing here represents a place in which there was strong Bragg reflections, and identify the crystals. So this is not a case in which they pre-located all of the crystals. They did have to do a scanning search here. I don't know if this could be modified to do a crystal pre-search or not. This is what it looks like. This is what it looks like mounted. And again, a very low, this is air, and this is their in-house thing. And these are some of the absorptions from other commercial devices in which in situ crystallization is. So I tried to get us slightly back on schedule. I hope I've given you a hint of some of the exciting things that are on the horizon and that would be good for us to keep our eyes on in terms of improving workflow. I'm particularly interested in pushing on with the electron microscopy thing. I think it could serve many users very well to know better about what's in our drops. Thank you.